All right, I chose CAR T cells for cancer treatment as my presentation topic, and I think this is a really important topic for everyone to learn about. Almost everyone knows someone who's been affected by cancer. Probably most of us even have a close relative. And so this treatment is sort of at the forefront of treatments right now as far as immunotherapies that are being heavily researched and developed. And so CAR T cell therapy is a form of ACT, which is adoptive cell transfer, and the CAR in CAR T cells stands for chimeric antigen receptors, which are the synthetic receptors that are grown on the T cell when they're removed from the body, and we'll get to that later. But basically, that antigen receptor will allow them to target specific proteins on the cancerous cells that normally they may not be able to target as effectively. So the current most promising method of altering T cells to be able to target an antigen is by altering them to be able to target B cells, and specifically an antigen found on them called CD19, which was one that we talked about in our quizzes in the past. So the altered cells are then expanded in a lab into the hundreds of millions and re-injected back into the body, revitalizing the whole immune system. And although it's not for very many cancers right now that this is being used, it's being heavily researched and developed and is going to be improving a lot in the next couple of years. And because they're using your own immune system, it's been referred to as the living drug. And so that's pretty neat. And so without these CAR T cells, our immune system would like to kill cancer, and our T cells are sort of the leaders of that battle against the cancerous cells. But there's just cases where they're not able to. And so they'll patrol our bodies looking for the abnormal cells, which is determined by the receptors, which are the little claw-like structures on the end of the blue cells there. And if they have a receptor compatible with the antigen, which is the red structure on the end of the red cell there, which the red cell here is a cancerous cell. If their receptors are compatible with that antigen, they can latch on and they can kill that cell without harming the rest of the body. And when they attach to that cell, they're able to send out a signal inviting all their other T cell buddies to come over and jump the cell. And that's much more effective than a one-on-one cell fight against cancerous cell and an immune system cell. So if the T cells don't have the receptors to recognize the antigen on a cancerous cell, there's nothing that can be done to fight it, and it'll just keep growing and growing and eventually kill you. Cancer can find ways to disguise itself from the immune system. They can be disguised as healthy cells, or like in this picture here, they can sprout too many antigens for the T cell receptors to attach to and fight them. And because they can't attach, they then won't be able to send out the signal to all their buddies to come fight the cancerous cell. They can also find ways to turn off the immune system's response against them, or they can quickly kill T cells that attach to them, preventing others from attaching to them, things like that. And so that's why CAR T cell therapy has been developed. And recent technology is really what has allowed it to be developed, probably starting in the early 2000s, because back in the 60s when they were studying immunotherapy, they could see all these, they knew immune system cells were fighting off deadly pathogens and diseases and all that, but they didn't know what they were, they couldn't alter them, none of that. So we'll get into that a little bit later with CRISPR and all of that. So for CAR T cell therapy, it's pretty simple to explain in layman terms. It's pretty simple to explain in layman terms, but it's probably much more complicated for the scientists. So basically, blood is drawn from a patient, and the T cells are separated out from the blood. And then a man-made inactive virus is introduced to the T cells, and it carries no disease. All it does is give the cell the information to develop the CARs. So step 3, the CAR T cells are now modified to produce chimeric antigen receptors, or CARs, on their surface, which are these yellow receptors on this blue cell here. And that will allow them to fight the cancerous cells. It'll allow them to recognize a protein antigen on the surface of the cancerous cells, or the deadly cells. Step 4, the cells are then modified into the millions in a lab and injected back into the patient. And then step 5, the CAR T cells are now able to latch onto cancerous cells and hopefully fight them off. So in 2017, the FDA approved this for treatment with children with acute lymphoblastic leukemia and adults with advanced lymphomas. And I haven't been able to really find other treatments that it's available for under FDA approval, but it seems like all the websites I went to, every institute saying like this is the forefront of their treatment and they're heavily researching it and it's really promising, and they want you to meet with a doctor to see about your situation and all that. So I think that's good news. I think that means it's in development and it'll be improving in the next couple of years, and hopefully it'll be available for more types of cancer. Some of the side effects from this treatment are cytokine release syndrome, which is similar to flu. You get headache, fever, chills, severe nausea, vomiting, diarrhea, muscle and joint pain, shortness of breath, low blood pressure, and a fast heart rate. And that seems like, that's terrible, but compared to what some of these people may have to go through if they didn't get the treatment or if the treatment wasn't available, I think they'd much prefer the muscle and joint pain to potentially death. So that's pretty promising. And then here, this is Dr. I believe it's Mikel Sadelain, and he's recently been showing that CRISPR can really be used to improve the way the CAR T cells are used. They can place the CARs, the antigen receptors, at a really specific location on the T cells that makes them much more effective, less side effects, faster, just all around more effective. And I'm going to show a little timeline here. All right, so you see as early as the 60s, they were able to see that immune cells were protecting mice from cancer, but they didn't know what the cells were, how to modify them, alter them, any of that. And if we go up here, I think it's 2002. So first effective CAR T cells were developed, and that man in the middle is Mikel Sadelain. He's the one that's right now still working with CRISPR and really at the forefront of this technology. So the second generation CAR T cells are able to survive, proliferate, kill prostate cancer cells in a lab. And that really established that this was a feasible thing to do if the technology could be developed and so on. And then all this is research. You can if you want to read all those, any of you can take a look at that. But in 2015, the FDA said that this is a breakthrough therapy. And then in 2017, like I said earlier, they approved it for some treatments. But here's Mikel Sadelain. He's been working with the genome editing tool CRISPR, which is something we've talked about all the way since, I think, the beginning of class, the potential that has to change medicine. And so that's really promising. There's not a whole lot more to it right now, but we're living right here in 2020, so hopefully there's more development on the way. And these are my sources. This top one is the primary literature. There's the abstract and intro and all of that. And then all of my pictures I got from this YouTube video. I just screenshotted it, and that's a really simple explanation. She probably explains it a little better than me, this person that made this YouTube video. But that's where I got my images from. I screenshotted that video. All right, thank you, and hope everyone stays healthy and has a good semester.